CrustNet.
The nitrogen fixation tubes are meant to collect intact soil cores containing the above-ground biocrusts and 1 centimeter of below-ground soil. One end of the tube is beveled to make it easier to insert the tube into the soil. We refer to these tubes as N-fix tubes throughout the CrustNet protocol, but they will be used for both carbon fixation and nitrogen fixation measurements in the laboratory. In Tier 1, you will collect N-fix tubes for the structure-function plots. Choose sampling locations that are representative of the organisms and cover of the plot. For instance, if the plot is 80% dark cyanobacteria, then you can collect 2 cores that are mostly dark cyanobacteria. As shown in the sampling diagram, avoid plot areas where you will make other measurements or take other soil samples. In the Tier 2 disturbance gradient experiment, you will sample at a standard location following the sampling diagram exactly. Before sampling, label the tube with the project name, the name of the node lead, the plot name, and the date. To core the sample, press the tube into the soil. You may wet the soil surface or use a mallet carefully if hand pressure is not sufficient. You only need 1 centimeter of below-ground soil, so stop when you have gone 1 centimeter deep. Then, use a knife, edge, a trowel, or other tool to capture the soil at the bottom of the tube and extract the tube from the ground. Next, push paper towels, toilet paper, or Kim wipes into the bottom of the tube to block the soil from falling out. Then seal it with a rubber stopper. Add a cushioning material to the top of the tube and then seal it with a rubber stopper. Transport these samples carefully to keep the soil cores intact. Keep them upright and vertical. These samples need to be completely dry before shipping, so take them back to a safe place and remove the rubber stopper from the top of the tube and the cushioning material. Allow the samples to air dry for as long as necessary for the cores to completely dry. You can use a fan or a hood with airflow to increase drying. Please reach out to the leadership team if you are not able to dry the soil cores. When shipping these samples to us, you can use other bagged soil samples to prop the tubes upright in the box, or you can use an interior box to ensure that the samples have extra padding and remain upright.